Hello dear friends, myself Dr. Kishore Kunal, and today we are going to see a very important topic from a very important chapter. So from this chapter approximately 4 to 5 questions are asked every year, and specifically from this topic minimum 2 questions will be asked. So this becomes very important topic. So today we are going to see this topic, and in this let's first start this by understanding what is central dogma. So let's first understand central dogma. So central dogma basically is that DNA will get converted into RNA, or from DNA RNA is formed through the process called transcription, and from RNA through the process of translation protein is formed. Now here this DNA can also undergo replication. So this is called replication. So these three important processes, replication, transcription and translation, form the central dogma. They are conserved in all the living organisms. So here we will mainly in this topic focus on this part, that is the transcription of DNA. But how is this transcription different from the replication? In replication we have already seen that the entire DNA segment will get copied. The entire segment of DNA get copied in replication, but when it is transcription, then not the entire part of the DNA, but some specific part of DNA will undergo transcription, right? So first difference is that the entire DNA will undergo replication and a part of DNA will undergo transcription. Now the second thing is that DNA replication will produce a double-stranded DNA, whereas transcription will produce a single-stranded RNA. So from a double-stranded DNA you get a single-stranded RNA. We will see how this single-stranded RNA is formed, but before that let's understand what is the single unit of transcription. So transcription takes place in the DNA, and as I said, a small part of DNA always undergo transcription. So the unit of transcription is called transcriptional unit, and this unit consists of three parts: the promoter, structural gene and the terminator gene. Now here if you see very carefully, this is the DNA, one strand, another strand. So these both strands are complementary to each other. Replication follows the principle of complementarity, that is, one strand of the DNA will be used to synthesize another strand of the DNA. In the same way, transcription also follows the principle of complementarity, that is, from the sequence of DNA you will form a sequence of RNA. And the difference between DNA and RNA remain the same, that DNA will have thymine as the nucleotide, and in case of RNA it will be uracil. So now here the transcriptional unit, as it consists of promoter, structural gene and terminator. So here if it is two strands, so DNA strands are anti-parallel. So as we can see here that these two strands are anti-parallel. Now out of this, one of the strand will call template strand, another strand will be called coding strand. Now how we will say that which strand is template strand? So the strand from which the RNA is synthesized is called template strand. Let's say our RNA is formed from this strand as the complementary sequence, as the complementary pair, then this strand is called the template strand. But the synthesized RNA will be, similar to the coding strand. So understand this, from 2 strand of RNA you synthesize one, from the 2 strand of DNA you synthesized 1 RNA strand. So the strand which you use to form complementary RNA is called the template, right? It is called the template strand, and since your RNA code will match mostly with the coding strand except the uracil part, so this is called the coding strand. So this point is very clear. Usually question is asked that coding strand 5 prime to 3 prime, template strand 3 prime 5 prime. This side is called upstream and this side is called downstream. Now in this, again few things you have to keep it in mind. Whenever I say upstream or downstream or any position in this transcriptional unit we explain with respect to the coding strand. For example, if I say promoter is present on the upstream, means with respect to the coding strand. Terminator is present on the downstream, means with respect to the coding strand. Is it clear? So now here DNA, double-stranded DNA will form one RNA. So in this entire process, one enzyme is used and that enzyme is called DNA dependent RNA polymerase. This enzyme will synthesize the complementary RNA. And this here, before getting into the detail of how they are synthesized, now one question can come in mind that why the RNA is formed only from one template strand? They can form from this strand also, right? So why the RNA is formed only from one strand, that is the template strand? There are 2 reasons behind it. So if I ask this question, why both strands are not copied? Why only one strand is copied? See, if both strands are copied, then there will be 2 problems. The first problem will be that, see, for the single segment or for a single transcriptional unit, you will have 2 sequences of RNA. For a single transcriptional unit, you will have 2 sequences of RNA. From one unit, one transcriptional unit. And the second problem will be these 2 sequences which are formed will be complementary strand will be formed. Complementary strand will be formed. Now if complementary strand, 2 complementary strand is formed, then won't they will pair with each other, and will not it form a double-stranded RNA, right? So this will lead to, if both the strand are copied, then it will not result in a single-stranded of RNA. It will result in a double-stranded of RNA, and then that is of no use. It will not be used for translation. So our purpose is to create a single strand of RNA. So now we will see one by one how this process takes place. But before going to that, keep it in mind, what is a coding strand, what is a template strand, which side the promoter is situated, which side the terminator is situated, which side we call as the upstream, which side we call as the downstream. So these all points you have to keep in mind. Now let's move on to the next part, where we will see this transcriptional unit which will form the RNA. Now here, before going further, let's consider these 2 terms are used, right? So when we say gene, and when we say cistron, so gene we also call as a segment of DNA, right? Now, but what is gene and what is a cistron? Both are quite interchangeably used, but there is a clear difference between gene and cistron. See, DNA can form RNA, but this RNA can be tRNA, this RNA can be rRNA, and this RNA can be mRNA. These are the three types of RNA, right? So, a gene is the segment of DNA which can form all these three types of RNA, right? Now, here this tRNA is not used in protein synthesis, right? It will help in protein synthesis, but it will not get translated into protein. Similarly, rRNA, it will not get translated into protein. What get translated into protein is the mRNA. mRNA will form polypeptide. mRNA will be forming polypeptide. So now comes the difference between gene and cistron. Very carefully listen. Gene is the segment of DNA which can code any of this, tRNA, rRNA or mRNA, but cistron is the segment of DNA which code for polypeptide. Right? So now the difference is very much clear, that a segment of DNA which code for a polypeptide is called cistron, but overall if it form mRNA, tRNA and mRNA, it will be called gene. So can we say this line, that all cistron are gene but not all gene are cistron? Means gene will, if we take cistron, so cistron is a gene, but not every gene is a cistron, because tRNA will not get translated into protein. The rRNA will not get translated into protein. Only mRNA will get translated into protein. This point is clear. Now, since we have talked about this cistron also, so now there are two terms used. One term is called monocistron or monocistronic, and another is called polycistron. See, monocistron means only here it will be present in eukaryotes. It will be present in eukaryotes, and monocistron, the coding region will get interrupted. Means in between the coding region there will be non-coding region as well. Whereas polycistron will be found in prokaryotes. And here the coding region will be uninterrupted, and they are found in prokaryotes, right? So this we will see a little bit in greater detail when we see post-transcriptional modification, but this point is again very important. Usually students get confused between these two. What is monocistron and what is polycistron? Monocistron found in eukaryotes. Here the coding sequence is interrupted, means there will be coding sequence, then some non-coding sequence, then coding sequence, then non-coding sequence. Whereas in case of polycistron, they will remain uninterrupted. There is no non-coding sequence. So this is the difference between polycistron and monocistronic. We have already seen the difference between gene and cistron. So now we have the background ready to understand different steps of transcription. So let's see that. So intron and exon, just that part which we just said, that the intron and exon will be the coding and non-coding region, non-coding sequence of DNA, which we will see in the post-transcriptional modification in a little detail. So now let's move ahead in the steps of transcription. So transcription is performed in 3 steps. The first step is initiation, the second step is called elongation, the third step is called termination. So initiation, as I said earlier, we have already seen that the transcriptional unit will have 3 parts: initial promoter, structural gene and terminator, right? So in the initiation, what happens? In the initiation step, the RNA polymerase, which is the enzyme which play very important role in transcription. This is also known as DNA dependent RNA polymerase. This RNA polymerase will go and bind to promoter. This binding of this RNA polymerase to the promoter, it will create a bubble, or it will break the double-stranded DNA, right? This will create a gap between the double-stranded of DNA, and this RNA polymerase will then start moving. Now this RNA polymerase work in 5' to 3' direction. RNA polymerase will work in 5' to 3' direction, and here the new unit will be attached to the 3' end. So here what we see, RNA polymerase is functioning in 5' to 3' direction. So initiation is taking place, the single stranded bubble is created. Now the RNA polymerase will start moving. Now here initiation part is favored by another factor along with RNA polymerase called sigma factor. So sigma factor has a very important role to play in initiation. RNA polymerase is solely having active role in elongation part. The RNA polymerase will start moving in 5' to 3' direction, and this movement will create RNA. So RNA, as I said, that this step, the entire transcription work on the principle of complementarity, means a RNA complementary to the DNA segment will be created. So here this RNA segment is being created here, and in the elongation the RNA polymerase will keep on moving and synthesizing the RNA in 5' to 3' direction until it reaches a terminator stage. As soon the RNA polymerase reach the terminator stage, the RNA polymerase will fall off. And why this RNA polymerase fall off? Because of presence of another factor which is called rho factor. So what you say, in initiation sigma factor has an important role, elongation is solely controlled by the RNA polymerase, and the termination is controlled by the sigma factor. So rho factor, RNA polymerase and sigma factor, they all three contribute to this entire process of transcription. So now in the end what you will get? You will get a single stranded RNA. You will get a single stranded RNA. Now if we compare both this process in eukaryote and prokaryote, so then there will be some difference. So in case of eukaryote, there is additional complication. Now let's understand that complication. In prokaryote, the RNA form will have all the sequence which is coding sequence. All the sequence which is coding sequence. there is no non-coding sequence. Earlier we talked about intron and exon. So the non-coding sequence is called intron and the coding sequence is called exon. So in prokaryotes, the RNA formed will have only the coding sequence. And this coding sequence you call as exon. Means no intron is present. There is no intron. So that is why in prokaryote the RNA synthesized is called mRNA and it doesn't require any post-transcriptional modification. In prokaryotes, no post-transcriptional modification is required. Whereas when we go to the eukaryotes, here as I said, eukaryotes are monocistronic, means the coding sequence are interrupted, means there will be, let's say this is the sequence, so in between some sequence will be non-coding, some sequence will be non-coding, which is I am drawing with a broader line. So let's consider this as the non-coding sequence. So the non-coding sequence will be called as the introns, which is non-coding, and the coding sequence is called exons. Now, since here there is all the sequence is the coding sequence, so no modification is required, so it will directly enters the translation phase. Here they directly enters the translation, but in case of eukaryotes, this non-coding sequence need to be removed. So that is why here the RNA synthesized is called mRNA, but here the synthesized RNA will not be called mRNA. It will be called hnRNA. hnRNA. Now hnRNA stand for heteronuclear RNA. Now this hnRNA will undergo post-transcriptional modification and this hnRNA will become mRNA, right? So I can write it like this: hnRNA will undergo post-transcriptional modification to form mRNA. So from here we can clearly see the difference in the product of transcription in prokaryotes and eukaryotes. So if I summarize that difference, in prokaryote the formed RNA is called mRNA. Why called mRNA? Because it has only coding sequence. It has no intron, so it requires no post-transcriptional modification, and since it do not require any post-transcriptional modification, so it directly enter into the protein forming phase, that is translation. Whereas here non-coding sequence is also there, so it will undergo post-transcriptional modification and it will not directly enter into the translation. So in case of bacteria, you will see that the transcription and translation going side by side, whereas here first the transcription will be completed and then only translation can take place. So now what are the post-transcriptional modification that we will see? So here, let's say this is the hnRNA which is formed. This we will call as the hnRNA. So here broadly 3 modifications take place. The first modification which is shown here. The first modification is 5' methyl guanosine capping. So on the 5' end of hRNA, 5-methyl guanosine is added. Now why is this added? To secure the 5' end, so that the nucleotide at the 5' end remains secured. The second modification will be, it is called poly A tail. Again, large number of A is added at the 3' end, and that will again secure the 3' end. Then you will say, why so much large number of A is added? Because the 3' end is more reactive. So there will be more deterioration on the 3' end as compared to 5' end. 5' end is comparatively stable, 3' end is comparatively unstable. So that is why a large number of A will be there to protect it. And then comes the third modification. As I said, this has both coding and non-coding sequence. As you can see here, introns are present, and so this is the non-coding part. So you have to remove the non-coding part, and non-coding part is removed through RNA splicing. We call it splicing of introns. So after all these 3 modifications, you get a mRNA. So it starts with hnRNA, and after these 3 modifications, it becomes messenger RNA. So mRNA will have a 5' methyl guanosine capping. It will have 3' poly A tail, and all the intron will be spliced. So now mRNA will have only coding sequence. There will be no non-coding sequence in the mRNA. Non-coding sequence will be present only in the hnRNA, right? So now we have got the mRNA. So now this mRNA will be used in next process, that is translation. And in that translation, the mRNA will be there, the 2 subunits of ribosome will be there, and then they will form the polynucleotide. They will form the polynucleotide. So this mRNA, this ribosome, 2 subunits are there, and they will form the polypeptides. So which will ultimately form the protein. So mRNA is forming protein by the process of translation. Now another thing you have to remember that, see, the transcription part is taking place in the nucleus. Transcription, we have seen the entire process take place in the nucleus. Then the translation part will take place in the cytoplasm. Transcription will produce mRNA, translation will produce polypeptide, which will form protein. So this is the entire setup through which a segment of DNA will ultimately form the protein, which include transcription and translation. So that is it for today. In the next video, we will cover how exactly the translation take place, what are the different steps of translation. So till then, keep on reading NCERT, keep on revising, and best of luck. Thank you.